Hello, everyone. I'm Laila El Khattabi. I am a clinical associate professor and the head of a genomics unit at Pitié-Salpêtrière hospitals in Paris, France. And today I'll be sharing exciting results from the French national prospective study CHROMAPS, which aims to evaluate the performance of optical genome mapping and long-read sequencing for the detection of structural variants. Please note that this work is a collaborative effort, and I'm presenting on behalf of our consortium. First, I have no conflict of interest to declare. Now, the rationale for this study stems from the observation that chromosomal aberrations in postnatal constitutional disorders are primarily seen in two main contexts. So reproductive disorders where causal sex chromosome aneuploidies and structural chromosomal aberrations affecting either autosomes or sex chromosomes are typically detected through karyotyping. In this context, the diagnostic yield is estimated to be between 5 and 10%, depending on the population studied. And the second context is developmental disorders, including neurodevelopmental disorders, where copy number variations, including aneuploidies, and at a lesser extent, structural aberrations that may alter gene expression, account for approximately 15% of diagnosis, as evidenced by DNA microarray analysis. The key question here is, can we improve these rates? So we designed the CHROMAPS study to address this point. This study compares the performance of optical genome mapping and long-read genome sequencing with standard of care testing, karyotyping, or DNA microarray in detecting chromosomal aberrations. Our primary evaluation metric is the percentage of samples in which class 3 to 5 variants are identified. Individuals from the first group underwent OGM in addition to at least karyotyping, while those in the developmental disorders group received both OGM and long-read genome sequencing alongside at least DNA microarray testing as a standard of care analysis. All OGM and long-read sequencing analysis were performed blinded and subsequently compared with standard of care tests. This multi-center study involved 14 inclusion centers across various university hospitals in France, of which 4 sites in Paris, Rouen, and Lyon performed OGM and long-read sequencing analysis. A total of 390 individuals were enrolled, of whom 11 were excluded mainly due to non-compliance with the protocol. Of the remaining individuals, 141 were in the developmental disorders group and 234 were in the reproductive disorders group. In the reproductive disorders group, karyotype results were available for 220 individuals, of which 9% showed an abnormal chromosomal formula. Among samples with the normal chromosomal formula, OGM results were available for 93% of them. Of these, 3 analyses failed and still need to be reprocessed. Interestingly, 16 samples revealed additional events. 6 were clearly pathogenic. 8 were VUSs, variants of unknown significance, and 2 were CNVs with incomplete penetrance and variable expressivity without a direct link to the condition and their investigation. Here you have an example of a pathogenic CNV showing a microdeletion on the Y chromosome, highlighted here in red in the CNV track. This CNV was responsible of azoospermia in 2 individuals. And here is 1 megabase deletion shown, highlighted here in red in the CNV track and shown in the optical genome maps here. This deletion encompasses the STS gene, which is responsible for hypogonadism in 1 individual. Now, let's take a look at the samples in which karyotyping identified chromosomal aberrations. These 20 samples display a wide range of abnormalities, including sex chromosome aneuploidies and structural variations. At the time of analysis, OGM results were available for 17 of these samples. The aberration was correctly identified in 13 samples, while 4 were undetectable due to centromeric or heterochromatic breakpoints of the structural variation. Now let's turn to the developmental disorders group. Among the 141 included individuals, DNA microarray results were available for 129, detecting class 3 to 5 CNVs in 16% of cases, with multiple CNVs detected in 3% of them, in 3% of the total cases. For samples in which DNA microarray did not identify a causal CNV, OGM data were available for 82% of cases. Interestingly, 9 additional events were detected in 9 samples, including 7 variants of unknown significance or pathogenic variants, and 2 incidental findings. Here is one representative example of what initially appeared to be a 1.9 megabase deletion of the NSD1 locus on the optical map, as you can see here on the patient blue map compared to the green reference map. However, closer inspection shows that the CNV pipeline right here, the CNV track, does not detect, at least does not call deletion, suggesting that this may instead correspond to a balanced insertion, probably of this locus into a highly complex region that could impede SV detection. So this case is still under further investigation. In this developmental disorders group, DNA microarray identified a VUS or pathogenic CNV in 20 samples. OGM results were available for 15 of these, showing 100% concordance with 1 complex case that was better resolved using OGM. Here is the case. 3 CNVs on chromosome 9 were detected with DNA microarray. First, a 520 kb deletion on the P arm, followed by a small normal copy region right here, then a 10.5 megabase duplication, and a 5.5 megabase deletion on the Q arm. OGM revealed more complex events occurring on chromosome 9, as evidenced here on the circus plot. On the right, the zoomed region. So in addition to the 3 CNVs, intrachromosomal translocation or insertion events were detected, as evidenced by the pink connections on the circus plot. Closer examination of the insertion events shows that the first event connects the normal copy region between the deleted and duplicated segments on the P arm to the large deletion on the Q arm, as you can see here. So here is a zoom in of the optical genome maps. This segment, the normal copy segment, is also inverted based on the labeling pattern in the optical maps. This suggests a balanced inverted insertion. The second insertion event links actually the duplicated region of the P arm to the Q arm downstream of the deletion. And the labeling pattern on the optical map suggests a direct duplicated insertion shown here. So this is the Q arm part. As you can see from this figure, this aberration, at least the insertion of the duplicated segment, was confirmed by FISH analysis, as shown here, with a red probe that targets the duplicated 9p segment and a green probe targeting the deleted Q arm segment. The derivative chromosome 9 is indicated by the arrow here, and it shows the deletion of the green probe and the gain of the red probe on the Q arm. However, the inversion event was too small to be analyzed by FISH. Now here is a teaser of the long-read sequencing results in the developmental disorders group. Please note that these findings are very preliminary and still require further work, as you'll see. To date, we have processed 120 samples with a target sequencing coverage of 15x, which is the recommended depth for SV analysis. Coverage exceeded 15x in 90% of samples and was below this threshold in 10% of them, remaining though within an acceptable range. So we applied several SV and CNV colors, followed by annotation with annote SV. And at this stage, our analysis has focused solely on CNV detection using the HiFi tool, which was developed for PacBio data. As mentioned, this very preliminary analysis revealed issues with undetected CNVs, including the recurrent 15q11.2 deletion and 15q11.2 q13.3 duplication. Additionally, we identified two more VUS variants of unknown significance or likely pathogenic variants, and further characterized the complex rearrangement on chromosome 9. This is a case of an additional CNV. It's a deletion of less than 200 kb on chromosome 11. And this deletion encompasses the KMT5B gene, which may be consistent with the individual's phenotype. This is the complex chromosome 9 rearrangement, where the coverage depth plot that I'm referring to with the cursor shows a deletion followed by a duplication on the P arm and a deletion near the 9q heterochromatic region. So these are indicated by the red squares for the deletions and the blue square for the duplication. Without going into much detail on all abnormal events, these figures show reads connecting distant regions on chromosome 9, suggesting insertions and also inversions, as was the case with OGM analysis, as well as additional smaller events indicative of chromoanagenesis in this case. To conclude, the initial analysis of CHROMAPS study results is quite promising, showing a 30% to 50% increase in the detection of class 3 to class 5 variants with OGM in both reproductive disorders and developmental disorders groups. As expected, structural variants breaking within highly complex repetitive regions remain a limitation, which may necessitate complementary minimal karyotyping in the reproductive disorders group. And long-read sequencing data requires further analysis, but already indicates the need for a multivariant color approach. With that, I would like to thank our funders, INSERM and Agence de la Biomédecine, all my collaborators across France who made significant contributions to this study, and Bionano and ONT teams for their valuable support. Thank you for your attention.